Have you ever really thought about what's in your food? I mean, beyond the label. There are all these invisible little molecules that tell a pretty fascinating story. Today, we're going to be chemical detectives, and our mission? To uncover one of food's most basic building blocks, sugar.
The specific kind of sugar we're on the hunt for is called a reducing sugar. Now, that's just the sciencey name for it. Think of simple sugars like glucose. That's the basic fuel that powers pretty much all of our cells. These sugars have a special chemical structure that lets them react in a really specific, and lucky for us, a very visible way when we put them to the test.
And this is our main tool for the job, Benedict's solution. It's this brilliant crystal clear blue liquid. On its own, it doesn't look like much. But when it gets near reducing sugar and you add a little bit of heat, everything changes. This blue solution is our detective's magnifying glass.
Okay, so how do we actually do this investigation? Let's break down the whole procedure. It's a classic lab technique that's actually really simple but tells us so much.
It really boils down to just 3 core steps. First, you get your sample ready. So you might crush up some food, mix it with water to make a solution, and pop that in a test tube. Second, you add an equal amount of our bright blue Benedict solution. And third, you add heat. The heat is what really kicks off the chemical reaction we're looking for. Okay, now this next part, the heating, is super important to get right. You've got 2 main ways to do it. The best and way safer practice is using a water bath. You just place the test tube in a beaker of boiling water. It gives you this nice, gentle, even heat. Now you could use a direct flame, but that comes with a pretty big risk. The liquid can boil super fast and shoot right out of the tube. So yeah, the water bath is definitely the way to go. All right, once you've heated it, this is where the magic happens. This is the payoff, where all our detective work comes together. We're not just looking for a change. We're looking at a whole rainbow of colors that tells us exactly what's going on. It's time to crack the code.
Yeah, the source material just puts it perfectly. What starts out as that clear blue solution goes through this awesome transformation. It'll get cloudy and green, then shift to yellow, and if you get a really strong positive test, you'll see a brick red precipitate. And that precipitate, that's just the solid stuff that starts to form in the liquid.
So what happens if, well, nothing happens? If you heat your sample with the Benedict solution and it just stays that same old clear blue color? That's your answer right there. That's a negative result. It means no reducing sugars were found. Case closed.
But if you see any change in color, I mean green, yellow, orange, red, anything, then bam, you found it. That's a positive result, and it's your confirmation that there's reducing sugar in that sample. Congratulations, detective.
And get this, here's the really cool part. This isn't just a simple yes or no test. The color actually tells you how much sugar there is. It's like a scale. A little bit of green means there's just a trace amount. Yellow means a low amount. And as that color gets warmer towards orange and that final brick red color, it's telling you there's a high amount of sugar. The whole color spectrum is basically a data readout. Of course, any good detective has to follow protocol. And in the lab, that means safety first. Always. So let's quickly run through the absolute must-dos to make sure the experiment is safe and successful.
Okay, these rules are totally non-negotiable. First, you gotta wear eye protection. Second, always use a test tube holder for heating. Never ever your bare hands. Third, and this is a big one, notice how the tube is pointed away from everyone? You never ever point that opening at yourself or anyone else. And finally, if you absolutely have to use a direct flame, you've got to keep that tube moving constantly to spread the heat out.
So we've got the what and the how down, but let's talk about the why. Why does this simple little color-changing test even matter so much in biology?
Right. It's easy to think this is just some neat chemistry trick you'd see in a classroom. But its uses are actually fundamental to understanding the world around us, and even what's happening inside our own bodies. So what's the big picture here?
Well, this test is how we analyze the nutritional content of food, for one. It's also an amazing way to show biological processes in action. For example, you can use it to prove that enzymes in your own saliva break down complex starches into simple sugars. You can literally watch digestion happen in a test tube as the solution flips from blue to red. How cool is that? It makes these big abstract ideas totally real.
Benedict's test